So before we jump into mitosis, let's start off with interphase, which is composed of three subphases, G1, S phase, and G2 phase. In all of those subphases of interphase, your DNA, or your genetic material, is going to be in a decondensed chromatin form, which here looks like a homogeneous glob of genetic material. And you'll also notice some little darker spots. Those are your nucleolus, or that is your nucleoli. So during interphase, your cell is going to have a high metabolic activity. It's going to be conducting its normal cellular processes, interacting with its environment, serving its function, open for use. Again, your DNA is in an uncoiled chromatin form. So here we have double-stranded DNA, and that's going to be depicted here as this blue coil. So this blue thread or coil is actually going to be wrapped around these purple things, these purple globs. Those are going to be histones. So histones are a type of protein. You could think of it, like when you wrap your power cord around a power brick, the histone is the power brick, and then, your DNA is that power cord. But imagine your DNA is, you've got an infinitely long power cord and many power bricks that you just kind of, wrap around in nodes. So one of these nodes, which is DNA wrapped around a histone, we call that a nucleosome. Then you have some, free cord in between, or linker DNA, which then, links your nucleosome to nucleosome to nucleosome to nucleosome. And so when DNA is kind of organizing itself, you'll actually form these nucleosomes kind of stack together, forming a kind of a thicker cord. So in a sense, your chromatin form isn't entirely decondensed, but it is still very decondensed compared to a condensed chromosome form. So here is your chromatin form, and we will talk about either in this section or the following section how what you could do to express the genes that are actually wrapped around these histones could be loosened up a little bit by acetylation or tightened up a little bit by methylation in order to conduct gene expression. All right, so again, chromatin is just going to be a gigantic linear double-stranded molecule of DNA complexed with proteins called histones. And that forms a dense material, but again, it's still kind of spaghetti-like. Now, when we eventually move into mitosis, what's going to happen is your chromosomes are going to get into this condensed form. So this is actually a depiction of a chromosome, you could say, like in metaphase of mitosis, for example, where you're going to have these sister chromatids linked together at their centromeres. So sister chromatids are two chromosomes joined together after they've been replicated and coiled. So, your DNA is going to replicate during S phase, and they won't appear to be these bilobed chromosomes until mitosis or mitotic phase. Again, you have this double-stranded DNA helix wrapped around these histones. We call these nucleosomes. You got the linker DNA in between, and then they kind of stack together to form a tighter coil. Now, this would be a depiction, so down here, this would be a depiction of a condensed chromosome during mitotic phase, whereas, chromatin would be more like this during your interphase. All right, yeah, and so here it just says these are metaphase chromosomes, those bilobed condensed chromosomes. All right, so jumping into mitosis, we'll start off with prophase. So prophase, I like to associate the P in prophase with preparation, because what happens is your nuclear envelope will actually dissolve, and your chromosomes, that's when they condense. So they actually become somewhat distinct when we're looking at it under the microscope. So here it doesn't look like a homogeneous mass. You could actually see various condensed chromosomes. So here again, your nuclear envelope dissolves, and your chromosomes will condense. So here it just says chromosomes appear condensed, nuclear envelope is not apparent. So prophase is the first phase of mitosis. I associate it with preparation. All right, so before we move on to the next phase, which would be metaphase, let's go over a few different terms. So, again, during prophase or during any part of mitosis or mitotic phase, your chromosomes are coiled and dense and visible and distinct from one another. Your nucleoli within your nucleus is going to vanish. That's going to happen, during your prophase. And then a few terms here, we have centrosomes. So these are going to be organelles often composed of two centrioles. So here, each big yellow guy is a centriole that assemble to form a spindle apparatus. And from that spindle apparatus, or that spindle apparatus is going to be, composed of spindle fibers. Those spindle fibers, which are depicted as these yellow lines here, are going to be composed physically of microtubules. Now the next term here is the centromere. So if we're looking at a condensed bilobed chromosome, you're going to have, each of these sister chromatids are going to have their own, centromere. But when we're looking at it under the microscope, they kind of look like they have a joined centromere. It looks like a single centromere because they're so close in proximity. Now those centromeres each have these regions called kinetochores. So kinetochores are regions that will eventually bind to those spindle fibers, and those spindle fibers are kind of what conduct or orchestrate the mitotic process. So they're going to push and pull on different organelles and your various chromosomes and pull them to one side or another by shortening in length. All right, so moving on to metaphase. So first we had prophase, then we have metaphase. So metaphase is the second phase of mitosis. I like to associate the M in metaphase with middle. And what happens is all of your chromosomes line up single file down the middle of your cell. So here it says, the thick coiled chromosomes are lined up down the center of the cell on what we would call the metaphase plate, and spindle fibers are attached to the chromosomes. So they're actually attached to the chromosomes by attaching to a region of their centromeres called the kinetochore. So it's attached to their kinetochores. Now after metaphase, we have a phase called anaphase. I like to associate the A in anaphase with apart. And what's happening is your sister chromatids begin to move apart from one another. So anaphase, the chromosomes have separated and are moving towards the opposite poles. So now you've split apart your sister chromatids, which are just duplicates. You could say duplicates of genetic material. These are, identical, hopefully identical pieces of genetic material. All right, so in anaphase, most of the motion of your chromosomes are going to come from your kinetochores. There are these molecular motors at the kinetochores, which gives you about 75% of the movement, and the other 25% of the movement, like I mentioned earlier, comes from the shortening of those microtubules, or you could say the spindle fibers at the poles. So they'll latch onto, your chromosomes, and then they'll shorten from the point of the pole, essentially pulling the chromosomes towards them. All right, so first we had prophase, then we had metaphase, and then we had anaphase, which moved things apart, and then lastly, within mitotic phase, we have telophase. So I like to associate the T in telophase with turn back, because what happens is your chromosomes begin to decondense or become more diffuse, and your nuclear envelope will actually begin to reestablish itself. So it'll reform. So in this case, each of your chunks of genetic material are kind of becoming localized here. The nuclear envelope will begin to reform, and your DNA will begin to decondense back into chromatin form. And then to seal the deal, we get a process called cytokinesis, which is just the cytoplasm actually splitting in half. So here you have this cleavage furrow forming during telophase of mitosis, and then eventually you get cytokinesis, which is the splitting of, that one larger cell into two daughter cells. So you get a cleavage furrow, and it's due to constriction by actin and myosin, which are types of proteins that are involved in constriction, or in you could say that they're components of your cytoskeleton, proteins that help compose your cytoskeleton. All right, so now if we're dealing with plant cells, it's not as simple as just splitting the cytoplasm. You not only have to split the cytoplasm, but you also have to form new cell wall in between those cells. And so what happens is you will deposit vessels between the two new nuclei, and those vesicles, sorry, did I say vessels? Vesicles will be carrying cellulose. So remember, cellulose is what's going to compose your cell wall. And then hopefully at the end of all this, you have two identical daughter cells. Looks like these are, probably some onion root tips or some kind of plant cell. As you can see, they're rectangular or square in shape.